Good morning, students. Today, our session will be covering the topic on post-translation modification, which is the types of post-translation modifications that occur to the proteins after they are synthesized in the ribosomes. So we're going to cover their types, the localization of the proteins, the compartmentalization and secretion of these proteins. This topic focuses on how proteins are chemically modified after they are synthesized and how these modifications regulate function, localization, and cellular signaling. So, under the post-translation modifications, is a number of modifications that occur to these proteins once they are synthesized either in eukaryotic cells or prokaryotic cells. But understanding this post-translation modification is essential because rarely proteins function immediately after translation. Instead, they undergo modifications that determine their structure, activity, stability, and intercellular destination. So, the session learning outcomes is to state the types of post-translation modifications of proteins, to describe each post-translation modification process, to list the pathways of protein cellular localization, compartmentalization, and secretion, to identify the appropriate proteomic method to analyze post-translation modifications. We have common terminologies that we need to understand before we dive into more detailed information. So, what is post-translation modifications? And it is usually abbreviated as PTMS small s. Is the process of covalently adding a chemical group to an amino acid in a protein after the protein has been synthesized. So this is in very simple words. And but the post-translation modification, they have a major contribution in the configuration of the structure of the proteins, turning them into secondary, tertiary, and quaternary, as we explained before. An isopeptide bond is an amide bond. It's a peptide bond between the side chain of one amino acid and the side chain of main chain terminus of another amino acid. So these are bonds between one amino acid and the side chain, which is the R group, of main chain terminus of another amino acid. Ubiquitous protein, this is present everywhere in the cells, in the body fluids of the humans, animals, and in the cellular components of the plants, and even the bacterial cell or the prokaryotic cells. The small protein called ubiquitin was so named because it is present in all types of cells, and its amino acid sequence is identical in all creatures, from insects to humans. That's why I call it ubiquitous protein. The chemical name is ubiquitin. It's a protein, small protein, and it plays a major role also as one of the methods of post-translation modifications to the proteins. Post-translation modification enrichment is a term used to, it's an analysis module part of the steps of analyzing a post-translation modification of a protein. If you are studying a post-translation modification of protein, you will come to through one of the steps of this process is what we call enrichment. So it's an analysis module allows the researcher to search for enriched methods to enhance the availability of a specific protein from proteins list. Example, you're using antigen antibody or Western blotting. All this as if you are selecting a protein from a pool of other proteins. So this a way of enrichment, either using an antigen, in this case the protein will be the antibody, or using Western blotting, which is a method used in the laboratory to also to react with specific proteins. It's like tagging certain proteins. And this one we're going to speak more when we come to the different methods used in proteomics. Chaperones or molecular chaperones are a diverse group of proteins that assist other proteins, newly synthesized or stretched proteins, to achieve their proper three-dimensional functional shape, which is native conformation. So these are assistant proteins, we call them, but they have a major role in assisting the proteins to fold and have their secondary or tertiary structures or quaternary structure. We continue on the common terminologies. So we have post-translational protein localization, and this refers to the regulated trafficking and retention of fully synthesized and mature polypeptides to specific subcellular destination. This destination could be the nucleus, the mitochondria, and the plasmic reticulum, or chloroplast, as in the case of plants. And this is usually mediated by covalent post-translation modification. So even the protein to reach its final destination, it has to be post-translational, pass through the post-translation modifications that is specific to a certain protein. So these mechanisms, of course, they operate after ribosomal release and folding of the proteins. Protein compartmentalization is the spatial segregation of proteins into distinct cellular regions or organelles so that specific sets of proteins occur in defined separated microenvironments inside the cell. This organization ensures that different or even incompatible biochemical processes can run in parallel with high efficiency and precise regulation by keeping the relevant proteins together in the right place and excluding others. So this is what we call segregation of the proteins, which are similar in structure and similar in function, in one place. So to increase the efficiency of these proteins in their function within the cell. The other terminology is the post-translational protein secretion. And this refers mainly to how already synthesized proteins are moved across or into membranes after translation is complete. Either they are excreted outside the cell or they are excreted from the nucleus, for example, into the cytoplasm, or from any of the organelles like in the plasmic reticulum into the cytoplasm or into the Golgi apparatus and so on. So this is how they move and what motivates them to move from one position to the other, and this is called secretion. So what is post-translation modifications? As we said, these are set of transformations or chemical transformations that help to diversify the limited genome of organisms. If you have a genome, and as you know, the genome, it carries the genes, and these genes are transcribed and translated into proteins in the ribosomes. Um, these proteins are the primary structure of these proteins is this is sequence of amino acids that give rise to the polypeptide chain. But uh, to diversify this polypeptide chain, they undergo different types of post-translation modification. So a single sequence can be post-translated differently, because to undertake different functions, because they have different functions within the within the cell. So, post-translation modification refers to the covalent and generally enzymatic modifications of proteins following protein biosynthesis, either in prokaryotic or eukaryotic cells. It's very important in proteomics to understand and to analyze these post-translation modifications. Is the base of proteomics, actually, because if you are talking about proteomics as um, understanding the function of proteins and making use of these proteins for any um, environmental integration program or projects or therapy production, drug production. So you need to understand the post-translation modification of a protein. So characterization of this post-translation modifications is very important as a cornerstone in proteomics studies. So usually these post-translation modifications, it's a process within the cell as in response to a variety of intracellular, extracellular signals. So several modifications take, for example, place within the cytoplasm and the nucleus, for example, like phosphorylation and acetylation. Other types of modifications takes place in the endoplasmic reticulum and Golgi apparatus, such as glycosylation, adding sugar residues to the proteins. So we are going to speak about the different types. There are actually a lot of, there are very huge number of post-translation modifications, but we are going um, uh, to um, to have more insight in common post-translation modifications that are very common, and that is found commonly in prokaryotic and eukaryotic, and most of the proteins, they undertake this pathway of post-translation modifications. So the purpose and importance of post-translation modifications refers to the, as I mentioned, is the chemical changes of proteins that might undertake after translation. So these modifications affect a wide range of proteins' behavior and characteristics, including enzyme function and assembly, protein lifespan. Even the degradation and the recycling of proteins is part of these post-translation modifications. Protein-protein interactions, cell-cell and cell matrix interactions, molecular trafficking, receptor activation, protein solubility, it's affected by post-translation modifications, protein folding, and protein localization. So the post-translation modification, it increases the protein diversity. Therefore, these modifications are involved in various biological processes, such as signal transduction, gene expression regulation, DNA repair, and cell cycle control. And I'm sure you had this in the genomics, how the DNA, the mutated DNA, is repaired by certain enzymes and excised by certain enzymes. These enzymes themselves are a response to post-translation modification. They undertake their function. So post-translation modification occur in various cellular organisms, including the nucleus, cytoplasm, in the plasmic reticulum, and Golgi apparatus, and in plants, also in chloroplasts. So proteomic diversity, as you can see, the protein starts as, once it is translated, it starts as simple polypeptide chain, but its complexity increases as it goes to the from synthesis to post-translation modifications. So post-translation are key mechanisms to increase proteomic diversity. So as we know, the genome, for example, in humans might comprise as 20,000 to 25 genes. The proteome is estimated to encompass over 1 million proteins, although, so the number of proteins that are functional is not um, proportionally equivalent to the number of genes. No, the protein, it can, as I mentioned, once it is translated, it can undergo different types of post-translation modification. And due to this difference, there is a diversity in the type of proteins. There is diversity in their conformation and folding. There is a diversity in their function, although their primary structure could be similar. So changes as the transcription and messenger RNA levels increases in size of the transcriptome relative to the genome, and the post-translation modification exponentially increase the complexity of the proteome relative to those to both the transcriptome and genome. So it's very clear how the proteome complexity increases with the post-translation modifications. So there are different types of post-translation modifications. A post-translation modification are divided into reversible and irreversible. The reversible reactions contain covalent modifications, and the irreversible ones which proceed in one direction, such as proteolytic modifications. So it's usually leads to the lysis of the proteins. That's why it's one directional. So as you can see from the diagram here, for example, we have the reversible modifications, post-translation modifications. For example, addition of polypeptides, like as I mentioned, ubiquitin, or protein conjugations, or for example, addition of complex molecules like ribosylation, ADP, adenosine diphosphate ribosylation, adding sugar residue like glycosylation process, isoprenylation. Also, this is all these types are reversible. It's because these are all covalent bonds that's linked to the polypeptide chain. Other type of reversible post-translation modifications is the addition of chemical groups like hydroxyl group, hydroxylation, phosphate group, phosphorylation, acetyl group, acetylation, methyl group, methylation. So all these three types are considered um, reversible. The irreversible types, example, the amino acid modification, for example, deamination or elimination. It's all these types of, and we are going to speak about, we are going to take, as I mentioned, the common types of from each group, the common types of post translation modifications, to have more details about it. Proteolysis, of course, it is irreversible since it is cleavage of the polypeptide. So all these types of post-translation modification is grouped into two: reversible and irreversible. The table here is it gives more insight on the different types. You don't need to know it by heart. It's just exploring the different resources that present the different types of post-translation modification. It's just for your knowledge. You don't need to know it in depth. So, especially if you want to do research, such tables can guide you. So, from the table here, you can see a lot of a number of post translation modifications that is occurring both in plants and animal cells, in eukaryotic cells in general. Also, we have the other table also continue to the first table. Also, it gives more insight about the mechanism um, that is occurring and the output, if it is in plant or in animal. So such tables are very helpful in planning your experiment if you want to move on with post translation modification analysis. The same thing in prokaryotics. There are different types of post translation modifications. As I mentioned, we are going to have more insight on this, but multiple protein residues can be covalently modified. For example, for phosphorylation, there are different types of phosphorylation depending on the type of amino acid that is present within the primary polypeptide. For example, phosphorylation can happen if there is a histidine, asparagine, arginine, serine. So it depends on the polypeptide. That's why we have different types of phosphorylation. But at the end, the added group or chemical group is a phosphate group. So, but it depends on the type, on which type of amino acid that is present within a specific polypeptide chain, within a specific sequence that is signaling phosphorylation. So again, the glycosylation also differs. Acetylation also, it depends on the presence of lysine, protein, amino acid in the termini. Glycosylation, for example, it depends on the different types of amino acid like asparagine, arginine, serine. It depends on their sequence within the polypeptide. But these three slides is just to orient you about the different types of post-translation modifications that happen in both the eukaryotic cells and the prokaryotic cells. One of the methods that used in prokaryotic cells, what's called the systems that's used in prokaryotic cells post-translation modification, is the prokaryotic pop protosome system. This is found in some types of bacteria, especially the actinobacteria, and this encodes a signal modifier protein that is functionally related to ubiquitin. And as I mentioned before, ubiquitin is a type, it's a short or a small length peptide that is attached itself to proteins, and it tags protein for degradation. So in prokaryotic, there's a similar system, but it's called this type of protein is called prokaryotic ubiquitin-like protein, or abbreviated as PUP. So PUP is a protein about 60 to 70 residues of amino acids, and it attaches itself to lysine of the side chain of lysine amino acid, targeting proteins via their side chain carbohydrate of its carboxyl terminal. That means the lysine that is found in the carboxyl terminal, and also the glutamate residue in a process called population. So the population is the process of post-translation modification that tags proteins for degradation. But the signal for this system is the presence of lysine and glutamate residues in the amino acids. So, of course, there are signals within the cells for tagging these proteins to be degraded. Otherwise, if it's functional protein with the same signal of lysine and with the same residues, it will not be degraded if it's functioning for a specific enzyme or a specific I mean it has a specific function within the cell. But of course each protein it has its life cycle. Once it is tagged with this pop, then should be degraded and it undergoes proteolysis, and this with certain enzymes, and usually there are enzymes that help in tagging in this process of prokaryotic pop proteasome system. And this is two enzymes, which is PAFA and D pipelase enzyme. This act differently on this process for degradation of the of the proteins that is non-functional within the prokaryotic cell. For the post-translation modification and the prokaryotic cellular physiology, as you can see here, the the post-translation modification prokaryotics, they regulate three important functions: metabolic adaptation, okay, and stress response, and virulence mechanisms. These are very important mechanisms, regulatory mechanisms. That is, as you can see here, they control transcription, they metabolic shifts and stress adaptation, control of all metabolism enzymes. And so the capsule and this capsule and biofilm formation, this is part of the virulence mechanisms. Anchoring of and export of proteins, this is also part of the virulence mechanisms. Virulence and pathogenesis, this is the overall function of the bacterial cells. So the post-translation modification is important for these three regulatory mechanisms: metabolic adaptation, stress responses, virulence mechanisms. That is different types of virulence mechanisms, and this helps the bacteria to rapidly respond to environmental changes and adapt to any environmental changes where it is dividing and increasing in number. So this is major summary of post-translation modification. So the clustergram indicate the frequency of each post-translation modification on different amino acids. So you can see The frequency depends on different amino acids. That's mean phosphorylation. It takes place in which amino acid, also indicating the division of the post-translation modifications into five clusters. So frequency of major post-translation modification. This is shows the frequency of the post-translation modifications, and shows the frequency of each amino acid that was reported as as a modified site. So these different diagrams, it gives more information on the which type of phosphorylation linked to each type of amino acid, and the frequency of the phosphorylation that is more occurring within the cells, and the frequency of each amino acid that was reported as a modified site, signaling site, as we mentioned. According to this, we are going to select our post translation modification, and we are going to have more insight about the highest frequency types of post translation modification. So let us first speak about the reversible post translation modification, which are three: addition of chemical groups, addition of complex molecules, and addition of polypeptides. So first we have phosphorylation. So protein phosphorylation was first reported in 1960, and it is the most widely studied post-translation modification. And this process is an important reversible regulatory mechanism that plays a key role in the activities of many enzymes, membrane channels, and many other proteins in prokaryotic and eukaryotic organisms. So phosphorylation target sites at tyrosine, histidine, proline, so specific amino acid residues. But this modification mainly happens on serine, thyrosine, and histidine residues. So what does it include? It includes transferring a phosphate group from adenosine triphosphate to the receptors residues by kinase enzymes. So this modification can change the function of proteins in a short time via one of the two principal ways: by allosteric or by binding to interaction domains. So allosteric actually it's a biological mechanism where a phosphate group can bind to a protein at a site other than its active site, other than the presence of the specific residues, and this we call it allosteric site. This will cause a conformational change that modifies the protein's activity, and this can happen within the cells. But usually in this case, the protein function will be changed. um, the right thing is that the phosphate binds to its interaction domains where these residues are found within the polypeptide sequence. So, but as an overall function of the phosphorylation of these polypeptide chains has a vital role in cellular processes such as replication, transcription, environmental stress response, cell movement, apoptosis, and even immunological responses. So it has been shown that disruption of this pathway of phosphorylation can lead to various diseases such as cancer, Alzheimer's disease, Parkinson's disease, and heart disease. So this shows that phosphorylation is a very important and has high frequency within the cells. So it's very important that it binds to the interaction domains for proper functioning of the protein. So as you can see here, this is how the phosphorylation modification takes place. So this modification is catalyzed by protein kinases and protein phosphatases. These are the enzymes that takes place within the phosphorylation process. The kinases transfer a phosphate group, okay, from ATP to the target protein by the establishment of phosphodiester bond. So it the reaction, the chemical reaction between the amino acid and the its and the phosphate group. It's a link through a covalent bond, which is a phosphodiester bond. So phosphatase hydrolyze the phosphodiester bond by releasing the phosphate group and restoring the acceptor amino acid in its unphosphorylated form. So the kinases enhance the or catalyzes the production of the phosphodiester bond, while the phosphatases break the phosphodiester bond to release the phosphate group. So that's why we call it reversible process. In in prokaryotic, beside kinases, there is two component systems mediated by histidine for asparagine phosphorylation. So in the in the eukaryotic, the histidine should be a mediator between the phosphorylation of the different domains within the polypeptide chain and the asparagine and the between the phosphate group and the asparagine for asparagine phosphorylation. So this is this is one of the mechanisms that takes place in the prokaryotic. So the histidine is an intermediate between this phosphorylation process of asparagine. Asparagine is, of course, it is one of the domain of this one of the amino acid sites that is phosphorylated. The two component system are linear signal transduction systems. So the signal is transmitted via histidine for autophosphorylation of the sensory kinase and aspartate phosphorylation. So the outcome of this, the histidine is a mediator in the phosphorylation of asparagine. So as you can see here, it is reversible with kinases enhancing the addition of the phosphate group and phosphatases enhancing the reversing the process of phosphorylation and releasing the phosphate group. So it's a reversible process. The other process is the acetylation. So the first acetylation process modification in protein was discovered by Alfrey in 1964, and since then this is recognized as addition of acetyl group, functional group, and is added on a protein substrate. Acetylation is catalyzed by lysine acetyl transferase, or KAT, for N-terminal acetylation, and histidine acetyl transferase enzyme for histidine acetylation. So from this, you can notice that there are two important signals for acetylation: the presence of lysine and the presence of histone in the amino acid in the peptide chain. There are three forms of N-acetyl N-terminal acetylation, which is called N-alpha acetylation, N-epsilon acetylation, and O-acetylation. The N-alpha acetylation is an irreversible modification. So this is out of this context of reversible post-modification, and the other two types acetylation are reversible. The three forms of acetylation occur on lysine, alanine, arginine, asparagine, cytosine, glycine, glutamine, methionine, proline, and serine, and thyrronine and valine residues with different frequencies. Although the acetylation is more reported in lysine residue. The N-epsilon acetylation is more biologically significant compared to the other two types of acetylation. So the N-terminal acetylation has an essential role in biological processes such as chromatin stability, protein-protein interaction, cell cycle control, and even nuclear transport and actin and other nuclear activities. Acetyl lysine is vital for cell development and would lead, and its dysregulation would lead to serious diseases such as cancer, aging, immune disorders, neurological diseases. So and other cardiovascular diseases. So it from this definition, then lysine acetylation is very important for the different cellular activity. But the opposite is in bacteria. Lysine acetylation is ubiquitous modification in bacteria. That means it is it doesn't define the functions and could lead to the degradation of the polypeptide stacked by is identified as ubiquitin. Ubiquitin, once the protein is stacked with ubiquitin proteins like lysine, acetylated lysine, then this is lead to its degradation. So this acetylation affect the protein structure, protein or DNA protein interaction, and cellular localization. So it's very important for the bacteria to rapidly adapt also to environmental changes. So as you can see, this is reversible, and acetyl transferases, as we mentioned before, they use cofactor for adding an acetyl group to the alpha amino group of lysine side chain, epsilon amino group, and we identified the different types of amino acids according to their carbon atom, into alpha, beta, epsilon, gamma, and so on. Whereas deacetylases remove an acetyl group on lysine side chain. So that's why we call it a reversible, due to the presence of mechanisms of addition and removal of the acetyl group. The third process is methylation. So methylation is a reversible also a type of modification. It's dated back to 1939 and occurs in specific type of amino acids, lysine, arginine, asparagine, and glycine, and other residues uh, in in target proteins. However, lysine and arginine are the two main target residues in methylation, at least in eukaryotic cells. So as you can see here, lysine it's important amino acid for post-translation modification, for acetylation, and now for methylation. So that's where the primary sequence of the amino acid, the primary sequence of the polypeptide chain can be diversified according to its post-translation modifications. That's why post-translation modification increases the diversity of the proteome within the cell. One of the most biologically important roles of methylation is in histone modification. You already had this histone DNA interaction and the production of the nucleosome in the nucleus. Already, this is definitely explained to you in the genome part. So histones are very important in the these histone proteins are very important for the chromosomes. So methylation is in histone modification when histone proteins, after synthesis of their polypeptide chain, are methylated at lysine, arginine, histidine, alanine, and asparagine residues. So this is type of proteins that needs to be functional in the product in the structure of the nucleosome. Histones, they have to be methylated at specific domains or specific presence of certain amino acids in their polypeptide chain. So methylation of proteins eliminates the negative charge on the protein and increases proteins hydrophobicity. So methylation on carbohydrate side chain eliminates negative charge and adds hydrophobic properties. So the N epsilon lysine methylation is one of the most abundant histone modifications in eukaryotic chromatin, which includes transferring the methyl groups from adenosyl methionine to histone proteins via methyl transferase enzymes. The most important thing you need to note from this detailed information is that methylation is important for histones. You don't need to remember the types of enzymes because we don't go into details, but it's just for your knowledge. But their importance in histones is important. You need to know. Um, so in eukaryotic, methylated arginine has been observed in histone and non-histone proteins. So the importance of this methylation process is very important for the different biological processes, ranging from transcription regulation to epigenetic silencing and heterochromatin assembly, since histone is protein is one of the targeted proteins by methylation process. Defect in any modification in this modification can lead to various diseases, of course, as cancer, mental retardation, diabetes mellitus, and other types of diseases. So this shows, of course, any histone uh, defect in its methylation process. Of course, it will lead to defect in the chromosome folding. It leads to DNA exposure to the cellular enzymes, its degradation, and this all lead to type of mutations that lead to appearance of chronic diseases. So methylation, transfer of methyl group from S-adenosyl methionine or SAM to histone proteins. So the source of the methyl group is the chemical component, is the chemical compound S-adenosyl methionine. It's a compound that is found in the cell and it's a source of methyl group. Some changes to SAH, which is S-adenosyl homocysteine. So, so it's progressive from mono to di and trimethylation of lysine. Side changes and mono and dimethylation of arginine side changes in histone and terminal tail regions. So it's not only one group of methyl could be added. It could be added as mono or di or tri, according to the type of protein and according to the function of this protein. And but at the end, the source is all from of the methyl group is S-adenosyl methionine, which is found within the as a chemical component within the cellular solutions. Hydroxylation is a reversible post-translation modification carried out by the enzymes known as hydrolases and has the utmost importance in cellular physiology. The targeted amino acids are proline and lysine. Again, lysine is another importance. And the target proteins are the collagen amino acids chain undergo hydroxylation to be hydroxyproline and hydroxylysine. So this type of hydroxylation or process takes place in the endoplasmic reticulum, and the principal residue that is usually hydroxylated beside lysine is the proline. So hydroxylation takes at the gamma carbon atom of proline, and this hydroxylation of proline is vital component of hypoxia response or by hypoxia inducible factors, especially in the pulmonary system or the respiratory system. It's very important, the hydroxylation of proline as an important component in hypoxia response. Lysine is also hydroxylated at the its epsilon carbon atom, forming hydroxylysine. So multiple units of hydroxylation can be added to the proline or the lysine according to the function of the protein. These reactions, they need iron to begin, also molecular oxygen and other enzymes, while vitamin C is also used to oxidize iron. So the presence of iron is important for the hydroxylation process. So hydroxylation of proteins prevents tensile strengths by allowing fibers to cross-link with the proteins. So that's why it's important in hypoxia, because all the lung smooth muscles within the smooth muscles within the lung, they need to be flexible so that they can respond to any hypoxia. So this shows the hydroxylation process with the enzyme catalyzed by the enzyme hydroxylase, both for proline and for lysine. The number of hydroxyl groups is it depends on the function of the protein and its localization within the cell. The other type of reversible uh, post-translation modification is the addition um, of groups. Okay, so we have the glycosylation is of residues like carbohydrate residues, and this is very important. Is a complex form of post-translation modification by adding ubiquitous and having a high degree of variety.